In this week's lab, you will be examining the properties of enzymes through a series of activities. This video tutorial is intended to provide you with the information about the specific enzyme that we'll be utilizing today in lab, and to provide you with some instruction for how to carry out the lab activity itself. The first section of this video includes a simple demonstration for extracting peroxidase enzyme. This is not something that you will be doing in lab, but it provides you with some background information about the enzyme of study and illustrates the accessibility of peroxidase for study. This enzyme can be readily extracted from several garden vegetables that you can find in your local grocery store. Vegetables with high concentrations of peroxidase include turnips, parsnips, and horseradish. This demonstration will show how we can extract peroxidase from turnips. We begin the extraction by shredding the turnip into small pieces. Add the shredded turnips to the blender, then add 100 milliliters of pH 5 solution. When you turn on the blender, the blades will work to burst the cell walls of the turnip cells. This will release all of the cell contents. The buffer provides a solution that will suspend all of the cell contents and keep them from degrading too quickly after they've been released from the cell. We'll blend the turnip and buffer mixture for at least 30 seconds to maximize the extraction. After blending, we will need to pour the extract through a filter in order to remove any large pieces of turnip. Once we have our peroxidase extract, we will test it to make sure it actually contains the enzyme. We have sets of pre-measured test tubes containing pH 5 buffer solution and another set containing hydrogen peroxide mixed with guaiacol to complete the test. Before all of the solutions are mixed together, they are colorless solutions. When the peroxidase extract is added to hydrogen peroxide and guaiacol, the peroxidase reacts with the guaiacol to cause a color change from colorless to brown. The rate of the reaction can be measured by using a spectrophotometer to measure the rate of this color change. If we mix the contents of the pH 5 buffer tube with the tube containing hydrogen peroxide and guaiacol, you will see that there is no reaction occurring. In this case, we did not add any turnip extract, so there was no peroxidase in the mix to facilitate the reaction. This is our control treatment. However, if we add 1 milliliter of our turnip extract to the buffer tube and then mix it with the tube containing hydrogen peroxide and guaiacol, you will notice that very quickly a change in color begins to occur. This color change tells us that the solution we created with the turnip extract does indeed contain peroxidase. If you take a look at both the control and experimental test tubes in close-up, you can see that there is a very distinct difference in color. The extract that we created is a raw turnip extract, which means that in addition to peroxidase, it also contains all sorts of other enzymes and substances from the turnip. When you begin your lab exercise in class, you will use a purified peroxidase extract. This means that the extract you will use in class contains only peroxidase. In the remainder of this video, we will provide you with some instruction for how to use the spectrophotometer to collect data that you will need during this week's lab. Every group will begin by completing the standardizing the amount of the enzyme activity. This will give you an opportunity to practice mixing your tubes and placing them into the spectrophotometer in the right amount of time so you can begin collecting data on the rate of the enzyme reaction. This will take some practice to get just right, so use this activity to get the process perfected for your group. It is best to assign roles to your group members. Assign someone in your group to be the mixer. This person will actively mix the tubes, prepare the cuvettes, and place them into the spectrophotometer. Another group member should be assigned to be the timekeeper. This person can help provide the mixer with instructions and will keep an eye on the clock. Once the two test tubes are mixed together, they will need to time how long it takes for this solution to be placed into the spectrophotometer and then they will need to announce when it is time to record the absorbance data every 20 seconds for 2 minutes. The third member of the group should be the recorder. This person will record the absorbance data every 20 seconds for 2 minutes. The activity in this video utilizes the Genesis 20 spectrophotometer. However, as you learned in week 1, our labs utilize 2 different models of spectrophotometers. If your lab station has a Genesis 30 spectrophotometer, you will need to make the appropriate adjustments to use that spectrophotometer correctly. If you need a reminder, please watch the tutorial video to review how to use the spectrophotometers. You should begin the activity by making sure your workstation has all of the necessary equipment. Once you have everything arranged at your workstation, you will need to use the table provided in your lab manual to determine what solutions you need to place in each test tube. Label your test tubes and use the dispensers in the lab to dispense the correct solutions into each of your test tubes. This step has already been completed in this video. When you are ready to begin collecting data, make sure that your wash basin is filled with water and a little soap so you can deposit dirty test tubes directly into it to reduce any staining. Then, set your spectrophotometer to 470 nanometers. It has already been determined that this is the wavelength that provides the greatest absorbance for this activity. The first tube in your mixing table will contain all of the solutions necessary for your blank cuvette. Remember that this cuvette will contain all of the substances except the substance you want to measure. In this case, it will contain everything except the peroxidase extract. You will pour the contents of this tube into the cuvette, place the lid on the cuvette, wipe down the outside of the cuvette with a Kim wipe to remove fingerprints and any solution you may have spilled, and then place it into the spectrophotometer. You will then blank or zero the spectrophotometer. When the display reads 0.00, you can remove the blank cuvette and move on to preparing your first pair of tubes for data collection. You will begin by mixing the 2 tubes together. As soon as the 2 solutions are mixed, your timekeeper should begin recording the time. Your mixer should be working to get the solution into the cuvette and into the spectrophotometer within 20 seconds. Once the solution is mixed, you should pour it into your cuvette, cap it, wipe down the outside with a Kim wipe, and place it into the spectrophotometer. Your recorder should then begin recording the absorbance data every 20 seconds for 2 minutes. Let's see that process again in close-up. Set your spectrophotometer to 470 nanometers. Prepare your blank cuvette. Wipe the outside of the cuvette to remove spilled solution or fingerprints. Place the cuvette into the spectrophotometer. Then press the blank or 0 button to blank the spectrophotometer. When the display reads 0.00, you can remove the blank cuvette. Mix your next 2 tubes together. This is where the recorder begins recording time. Pour the solution into the cuvette, cap it, wipe down the outside of the cuvette, and place it into the spectrophotometer within 20 seconds. Then your recorder should begin recording absorbance data every 20 seconds. After mixing each pair of tubes and collecting your absorbance data, make sure that you rinse the cuvette and lid with water and dry them the best that you can before moving on to the next set of tubes. When you have completed your entire lab activity this week, make sure that you wash all of your test tubes and place them in the rack upside down so they will dry for the next section. Clean your workstation to return it to its original condition, and make sure that all of the necessary supplies are available at your workstation for the next group of students.